presentation?
Yes, we're good to start.
Okay. Before we begin, thank you very much to Ashok Potiani, Dr. Cecilia Colauti, and the organizers for the invitation to participate at this webinar. So as a matter of time, I'm going to try to be brief. What I wanted to talk to you today is about nanotoxicology of carbon nanotubes. So we have different speeches in which some of the topics were talked already. But I wanted to talk to you about the nano safety and how important for us is to address or to start to address how much harm the possible toxicological effects can do to human beings, mainly in workers who are exposed to direct processes of the manufacturing of these elements. Okay, so let's make a brief comment about some basic concepts. So we know that nanotechnology is the science that studies manipulation matter on a near atomic scale that goes from 1 to 100 nanometers to produce new materials and devices with the ability to transform many industries and their products. That goes from the medical industry, as we've seen in the speech before, to manufacturing like textile industries. So we know that nanomaterials are substances, materials that can be natural or manufactured in soluble ambient resistance with one or more external dimensions, or at least an internal structure in the range scale between 1 to 100 nanometers squared. So below we can find a picture which shows a comparison of elements at a nanometer scale. We can see a tennis ball on the right side in comparison and contrast to the size of a length of a molecule of water. And this is important. This is important because if we take an element or an object in a metric scale and we extrapolate that element or the same element to the nanometric scale, their physical chemical properties change totally. So that's why it's really important to address this, and the toxicological effects change too. Let's make a brief search through the history, really short historical overview, because sometimes it is thought that nanomaterials are new or novel elements, but they have been with us through the history of the human beings. One of the first examples that we can see in history is the Mayan mural of the city of Catzla. Scientists discovered a pigment known as indigo from genus Indigofera, and that was thanks to the impact of a light beam of a photoresection with dominant wavelength between 420 to 250 nanometers. Another example is the glass of Lycurgus, which is composed by nanoparticles of silver, gold, and copper up to 100 nanometers in size in a glass matrix. The other example is the stained glass window or vitrol of the Cathedral of Chartres, which is composed of nanoparticles of cobalt oxide. And what they share in common is this phenomenon called plasmonic excitation, that all of you might have probably heard already. And what this phenomenon causes is that when you impact with a light beam, the electrons that are on the most superficial layer interact between them, so they make changes in the color of the element that this light is projected. As a matter of example, this is the cup of Lycurgus, the one we see down here, and if we impact with an inner light, the cup becomes red, and if we impact with the outer light, it becomes green. This is thanks to this plasmonic excitation phenomenon. Moving forward, we have the refluid, which was discovered by Faraday and Tyndall, and then gave the name of the Faraday-Tyndall effect. And it is a colloidal fluid which is composed of gold nanoparticles, and they have a lot of properties. Moving forward to the modern history, nanotechnology was first coined by Norio Taniguchi in Tokyo in 1974, but the first conference about this science was given by Richard Feynman, who won the Nobel Prize in Physics at the American Society of Physics in 1959. Then the Buckminster fullerene, or the fullerene C60, was discovered in 1985 by Ron Kerr, Harold Kroto, and Richard Smiley. They developed this, and they won, thanks to it, the Nobel Prize in Chemistry in 1996. And lastly, this is moving forward to the matter of the topic of this conference, carbon nanotubes were discovered by or developed by Sumijima in 1991, and the first one that was developed was a single-wall carbon nanotube. So we know that carbon nanotubes are nano-objects with two external dimensions similar to nanoscale, and a significantly larger third dimension composed of carbon. They can be made up to one or more layers of graphene. We've had all the speeches about graphene and about carbon nanotubes, so I'm moving forward a little bit faster. We can see carbon nanotubes, carbon nanofiber, carbon fiber, and as I told before, depending on the amount of layers of graphene, they can be classified as single-wall carbon nanotubes, double-wall carbon nanotubes, triple-wall carbon nanotubes, or multiple-wall carbon nanotubes. And we know that nanoparticles share common characteristics in terms of physical-chemical properties, but at the same time, each nano element has specific physical-chemical properties. In the case of carbon nanotubes, we can talk about the thickness, which is given by the number of graphene layers or the clarity of the tubules, the longitude, which is established by the length of each CNT, dependent on the synthesis process and the metal catalyst. Then we have structure, which is caused by the arrangement of the graphene sheets, depends on the chirality of the tube. We can have a chiral zigzag or armchair atomic structure, but not only the manufacturing processes gives physical-chemical properties to the CNT, but also the defect that, of course, thanks to the manufacturing process. For example, the pentagon pair, the heptagon 5-7, and the bamboo-like structure. Then we have the surface to mass ratio or specific surface, which is given by the diameter, number of graphene layers, degree of grouping of them. For instance, the average surface area of a single wall carbon nanotube is 1,300 meters squared per gram, while the diameter of a kit that composes it is up to 6 times smaller, approximately 300 meters squared per gram. The bulk density, which is dependent on the CNT production process. For example, the bulk density of a crystal graphite is 2,200 milligram per centimeter cube, as opposed to graphene powder, which is nearly 200 to 600 milligram per centimeter cube. Then we can see the physical strength that was mentioned before. The tensile strength up to 200 G Pascal and elastic modulus, which goes up to 1 terapascal. To put it in other words, this means that CNT carbon nanotubes can be up to 10 times stronger than steel and 20 times stronger than steel and 20% stiffer than diamond. They also have electrical properties, can be conductive or semiconductor properties, which is directly proportional to its chirality. For example, CNTs have an electric current transport density that can be up to 1,000 times more than copper, which is really amazing. Then we have optical properties. So single-wall carbon nanotubes easily absorb near infrared light, so they can be used as a possible way of phototherapy. They have thermal properties which make them high thermal conductivity and high thermal stability, so they resist really high temperatures. We have to talk about generation of dust or dustiness. We're going to talk a little bit further about this later on, but it varies depending up to 70% depending on the manufacturing processes and the conditions of the work environment. Then we have the hydrophobic properties, which is the tendency to carbon nanotubes to form agglomerates. And finally, the functionalization, which is a post-production process in which we can make some changes in the outer layer. So the properties, either physical or chemical, can be changed, okay? So the bonds, like non-covalent bonds or covalent bonds, can be changed too. So I'm going to briefly describe just the different synthesis processes that we have nowadays in MARC, but which are the laser ablation, the arc flash discharge, the plasma torch or plasma cutting, and the chemical vapor deposition. But I'm not going to talk specifically about the process of each of them, but I wanted to give a approach because it's important that depending on the synthesis process, the dustiness or the amount of dust that it is produced will totally vary. So the exposure of the workers, which are directly near these work environments, okay? So we also have mass sampling methods, which are the scanning electron microscopy, the transmission electron microscopy, which studies morphology, dimensional radial, longitude, and tubule diameter of the TNT, the near-infrared spectroscopy, which analyzes the hydrodynamic site of the TNT in an aqueous medium, the thermogravimetric analysis, which studies the crystallinity and diameter of the TNT, and the Raman spectroscopy, which studies the composition of the mass of the TNT, like we've seen in the older speeches before me. These are one of the most important and used methods nowadays. We also have personal detection methods like direct reading instruments such as the condensation particle or the optical particle counter. We have sampling methods that can be area filters or personal use filters. And finally, thermal exposure rubber gloves, which can be studied at the end of a work day to see how much dustiness was the worker exposed to. So why is this important? Because when we develop a product, when we manufacture an element, it is important that we establish recommended exposure limits, okay? So, and this is primarily important because when it comes to carbon nanotubes and nanomaterials, a study conducted by Giuseppe Cano et al. In 2016, which analyzed more than 10 scientific studies that sought to measure the exposure of workers in more than 10 nano industries, which were located in Japan, the United States, South Korea, among others. They concluded that the areas where there was a greater exposure of workers were where there were no control measures implemented. They found traces of elemental carbon particles as well as CNT and carbon nanofibers, which were detected in aerosolized samples from the personal breathing zones that were taken from the nostrils, oral and perioral cavity of the workers. Okay, they found mainly single-wall carbon nanotubes and multiple-wall carbon nanotubes, and concluded that the process that has a lot of phases, such as the mixing, transferring, sieving, proving, weaving, and synthesis of the CNT, they found that the synthesis process of the CVD, the chemical vapor, and the discharge methods were the one which had the most percentage of dustiness production in comparison to the other methods. So we have international institutions which determine the recommended exposure limits. We can mention some of the more important, the British Standard Institution, the Dutch Social and Economic Council, and NIOSH and OSHA, which is the most used nowadays. And it says, or it stands for that workers cannot be exposed up to more than 1 microgram per meter cube of TNT and CNF up to 8 hours of weighted average time of respirable elemental carbon fraction. Okay, they cannot overpass this limit of time and amount of TNT and CNF. We can talk about the clearance and biodegradation. It was talked about before in the previous conference too. So it has some determinants. The species, for instance, whether it is a human or an animal, that we can see that the soluble or insoluble character of the nanoparticle, the dimension and length, which is similar to asbestos fibers, so they can reach the lower alveolar regions easily. The interaction with the phagocytic mononuclear system, and the property of direct translocation, blood or lymphatic route, they can reach to or access to organs such as the brain, spleen, liver, kidneys, and heart. and the recognized nose to brain route or pathway that we'll be talking about later on. So what NIOS found is that nanoparticles have an estimated deposition efficiency of up to 99% in the human respiratory tract, including the alveolar region values up to 60%. So they have a really good clearance and biodegradation in the respiratory tract. So we have several studies over here which talk about different toxicological effects of the carbon nanotubes. But in order to make this easy to see, we have a picture over here. But I wanted to let you know that nowadays we don't have studies that have used human being models, of course. Just murine models or mice models are being used nowadays. So what it is done is to extrapolate these possible toxicological effects to human beings, but we don't have human beings models nowadays. But the more important effects that they were found were, for example, hematological effects, such as activation of the classical complement pathway, affinity for purinogen and apolipoproteins, platelet activation and aggregation, hemolysis, cardiovascular effects, such as aortic mitochondrial activity damage, the acceleration of the progression of atherosclerosis, tissue activation, and pulmonary cardiac and aortic oxidative damage. We also found, you can also find effects on fertility in murine, male murines, which are reversible at last, partial damage to the seminiferous tubules, reduction of the germ layer thickness and spermatogonia, vacuolization of tertiary cells, degenerative and necrotic changes, vasodilatation and testicular hyperemia, which is inflammation of the testicles. We can also find central nervous system effects such as oxidative stress with neurodegenerative potential. That's what nanoplastics are being studied for. Something that Dr. Cecilia Glauti mentioned in her speech, which is really important nowadays. Activation of glial and pro-inflammatory cytokines, transient and chronic local inflammation. Although effects found were pulmonary effects, which versus the production of oxygen-free radical and KRAS oncogene activation that leads to cancer, increased fibroblast cells positive for labeling with TGF, beta, PSMAD2, and beta-catenin, which are markers. They found augmented incidence of bronchoalveolar carcinoma and adenomas, and pleural mesothelioma. Also, we can mention the gastrointestinal effects, which is the higher incidency of peritoneal mesothelioma, and finally teratogenic effects such as agenesis and hypergenesis of the limbs, hypodeformity, hip deformity, reduce or absence of the tail, fusion, lack of fusion, of bodies and vertebral arches, and increased size and weight of the lung and tracheobronchial tree. So in order to picture them a little bit more, we can see some studies in which we can see how the nanoparticle, how the carbon nanotubes interact. They activate the several cells from the immunological system, such as the B cell, the macrophage, the mastocytes. They liberate interleukins, and they cause inflammation in the lungs. We have neural effects. We can see that CNT can cross endometrium, placenta, the yolk cell, and they access directly to the fetus, causing oxidative stress and inflammation, which leads to the picture below, which is abortion, and teratogenic effects, like I mentioned before, the lack of fusion of the body of the vertebrae and the lack of fusion of the arc branches. We can see cardiovascular effects. Once again, oxidative stress that causes inflammation and DNA damage, which leads to atherosclerosis, acceleration of the atherosclerosis process, and finally thrombosis, hematological effects, which finally causes inflammation, thrombosis, hemolysis that leads to anemia, neurological effects. We can see here a picture that's the neuron brain, and we can see some black dots over there, which are agglomerates of elemental carbon and multiple wall carbon nanotubes that gather together. That's one of the physical-chemical properties that they have. And we can see that what happens is that it causes inflammation and destruction of the brain tissue, so it leads to acceleration of the neurodegenerative process of the brain. And finally, we need to mention the nose-to-brain pathway, which was discovered or first talked about in 2019 by Igeo Uchewu et al. And why is this important? Because if we take an element, for example, a biological agent, such as a virus or a bacteria or a parasite, a fungus, or if we take a silica, for example, or any metal, and if we take that in a metric scale, in order to access to brain, it has to be enter through our nose cavity, then it has to go to the trachea, then access to the lungs, finally to the alveolar region, and then it access to the bloodstream, finally reaching to the brain. But the carbon nanotubes and nanoparticles are so small. We know that they are so small that they can access directly to the brain through the axons of afferent neurons that are located in the brain and which are connected to the olfactory bulb, which is in our nose. So they access through this called vasa vasorum, which is the blood vessels that gives oxygen and nutrients to the neuron. So they access through this little thick bloodstream, and they go directly to the brain, causing the effects, the neurological effects that I mentioned to you before. So just to stick to the time, and in order to conclude, carbon nanotubes represent the advent of novel elements that open doors for problems in the future, like we are experiencing right now, in terms of the use of new technologies that improve the quality of life of men or human beings. Animal studies demonstrate multiple multi-organ pathological effects with regard to exposure to CFT. However, up to date so far, we do not have incident prevalent data of pathologies in exposed workers or human beings. Therefore, it is vitally important to continue their research and experimentation, as well as to establish protective policies for workers and their families. That would be all. Thank you very much to all for sharing your time and your attention with me.